it's Mrs. Horseman again, and today we're going to be talking about the second part of protein synthesis. So we're going to be specifically concentrating on translation. So just a few things I want you to remember. Proteins are made by stringing together or putting together these really long chains of amino acids. And the sequence of those amino acids determines what the protein is and the function of the protein. And they can be just, a few, like maybe 100 amino acids long, or they can be thousands of amino acids long. The order of that amino acid is specific to that protein. If you change the order of amino acids, you actually are changing the shape of the protein and therefore changing the function of it or its job, okay? We also talked about last time the language of how mRNA instructions is the genetic code. So the sequence of DNA, the sequence of nucleotides in a DNA strand determines the genetic code or the instructions for the mRNA, okay? Remember also that the genetic code is read 3 nitrogen bases at a time, and we call that a codon. And 1 codon, so every 3 bases, is equivalent to or codes for 1 amino acid. Okay, so that was just a quick review. It's really important that you know that information before we get started with the next part of it. All right, I just wanted to show you, this is a chart. You can see there's a couple different ways that you can read this chart. So if you have a square chart like this, when you're reading the mRNA strand, you go 3 bases at a time. So this would be the first set of bases. Then you would come over here to find the second set of bases that match it, and then the third set of bases are over here. So just for instance, if we had a codon that was CAU, we would come down here to the C, so we're going to start here, and then we would come over to the A box, so where the C and the A intersect, CA, and then the U is across this way, so that would be the amino acid that would code for would be histidine, okay? So there's a square chart. We also look at circular charts. That's actually what I prefer, and that's what I'll be showing you in class, but I wanted you to be aware of that. If you ever see a square chart, you can use both of them. So let's consider the following RNA sequence: UCC, GAC, GGU. Okay, we would read that sequence 3 nucleic acids at a time, or 3 bases at a time, so we would break it up into chunks. Those 3 codons are going to represent 3 different amino acids. So even though if you count, there's 9 bases there, there's 9 nitrogenous bases, it's actually only going to code for 3 amino acids. Okay, so from your chart of amino acids, you need to figure out which amino acids are going to be coded by those codons above. So from this set of 9 nitrogenous bases, or set of 3 codons, what protein is going to be made? Hopefully, if you used the chart in your notes, you came up with serine, histidine, and glycine. We'll practice this a lot in class, so if you didn't get it this time, that's okay, don't worry. We're going to keep practicing. Okay, so because there are 4 different bases that are read in groups of 3, 4 to the 3rd power is 64 possible codon combinations, okay? So there are 64 ways to read for amino acids, is another way of saying that. There's a few very special codons. The codon AUG codes for methionine, and methionine is the start codon. So every single protein is going to start with the amino acid methionine, okay? It's going to be found at the very beginning of the mRNA strand. It's going to tell the ribosome where to attach the mRNA strand. So it's kind of saying, hey, I'm right here. This is the beginning, okay? And I compared that before. If you're reading like an English paragraph, you would know where to start the paragraph because it indented, or the sentence because that was the capital letter of the sentence. There are 3 different stop codons that don't code for any amino acids, but it's just telling the ribosome, you're done. You can release now, okay? And at this point, it's the end of the protein. The ribosome disconnects from the mRNA strand, and it's available then to code for a different protein at that point. So let's talk about protein synthesis. Specifically, we're talking about translation this time, okay? So the creation, synthesis means to make something. The creation of proteins is called translation. The cell has to translate the language of nucleic acids into the language of amino acids. So just like when you're going from English to Spanish, or English to German, or English to French, or Spanish to English, or Mandarin Chinese to Arabic, or whatever, you have to translate that language. It goes between languages, okay? So similar to that, we're going from nitrogenous bases in DNA and RNA to amino acids in proteins, okay? There's a little bit of a different language. There's kind of a language barrier there. They're not the same thing. The site of translation is the ribosome, and we've talked before about how ribosomes are where proteins are created, okay? That ribosome has a very, very huge job. You know that proteins are so important to your body, and the ribosomes in every single cell are responsible for making sure those proteins are made in the right way, okay? So the ribosome has to make sure that the amino acids are linked together in the right order to make that protein. If you change one amino acid, you change the whole entire shape of the protein, and if you change the shape of the protein, you change the function of it, or the job that it's able to do. And that's probably a bad thing. So let's talk specifically about the steps that happen in protein synthesis. We know already that the DNA makes a copy onto the mRNA strand, okay? The mRNA strand then is going to leave the nucleus and it's going to go to the ribosome. That is transcription. Translation actually begins when the mRNA attaches to the ribosome. So we have our ribosome, it's in two pieces, it kind of looks like a hamburger bun. So the mRNA, which is like the single strand piece of nitrogen bases, is going to come and attach to the ribosome, and the ribosome is going to come together, and it's going to actually begin at that point, okay? It's going to read the mRNA strand 3 bases at a time. So every codon of mRNA is going to move through that ribosome. The proper amino acid is going to be brought to the ribosome by the matching tRNA. Remember we said tRNA is transfer RNA, and it's responsible for bringing the amino acid to the ribosome. It's going to bring the supplies to the building site, okay? The ribosome is going to stick that amino acid together with an extremely strong, it's called a peptide bond. It's just another kind of bond that makes those the electrons of that molecule stick together. It's going to stick those together with a really strong bond, and it's going to continue to do that in order to make the protein. Okay, so a little bit more information about tRNA. If you see here, the function of tRNA, we said, is to transfer amino acids from the cytoplasm of the cell, where there's like a huge store of amino acids, to the ribosome, where those amino acids are going to be used to build something, okay? On one end of the tRNA are 3 bases, which we call an anticodon, and you can see here there are the 3 bases that are at the end of this loop, at the end of the T. Those are going to be complementary to the codon that is on the mRNA strand. So we actually call that an anticodon. The tRNA, each one is a little bit different, okay? So every one has a specific amino acid that it attaches to. So if you look here, AAG is very specific to one particular amino acid that will link right up here. Okay, so you can see this like grayish zone. It's not really gray, they're just doing this in the picture to clarify. But right at the end of this gray piece is going to be the amino acid up here. That amino acid is very specific. This tRNA molecule can only pick up one type of amino acid, and that is determined by the 3 nitrogenous bases that are at the bottom of the tRNA molecule, and those are complementary to or match a codon on the mRNA strand, okay? So because it's complementary, we're going to call it an anticodon. So for example, if the mRNA codon is UUC, this would translate the amino acid phenylalanine, and I know that because I used that chart that we're going to practice using in class. The tRNA molecule that's complementary to this will have the anticodon. It's going to be complementary. So if we have the mRNA codon here, UUC, the matching complementary anticodon is going to be AAG. And it's going to carry that specific phenylalanine amino at the end of it to the ribosome. Every tRNA is used repeatedly, so it's going to bring the amino acid to the ribosome, drop it off, and it's going to go back and pick up another phenylalanine, and it's going to continue that process over and over and over again. Once its amino acid is left on the growing chain of amino acids, so on the growing protein at the ribosome, it's going to go back to the cytoplasm, go back to that store of amino acids that your cell already has, and it's going to find another one that's specific just to that tRNA molecule. All right, the codons have to be read correctly and in the correct order. So let's look at this statement. The red dog ate the cat. If you notice, all of these words are 3 letters long, just like the codons are 3 nitrogenous bases long, okay? If the code is read in the wrong, like it starts reading it in the wrong place, so it doesn't start at the very beginning, we could see instead of starting right here at the T, it's going to start at the H instead. It totally changes the message. Her, ed, oga, tet, heck, at. It's a ridiculous statement, okay? The result is gibberish. So the ribosome doesn't understand that though. So the protein is going to still be put together, but it's going to be put together by putting the wrong amino acids in order. So most likely this will be a non-functioning protein because the mRNA strand, the message, which should be like this, was misformed and it ended up like this instead. Okay? So let's think about this. If the sequence on the DNA molecule calls for a protein with the following DNA codons, we need to figure out the mRNA strand or the sequence of the mRNA nitrogenous bases, the sequence of the tRNA strand, and then we need to figure out what would be the amino acid sequence of the protein being made. Remember, when you're using your chart, you're actually using the mRNA strand, okay? So first we have to figure out what the mRNA strand is going to be. We have for our DNA TAC, TTA, CAA, ACC, ATA, ATT, okay? If you're looking at a DNA strand, it's actually all continuous. I separated it out because we know that the mRNA strand is read in groups of 3, and it's actually easier for our brain to look at things in sets of 3 than it is to be like a continuous strand. So that's why I did it that way. So if we were going to code the mRNA strand, I'm going to give you just a second, pause and see if you can figure out what the complementary strand of that would be. Remember, there is no thymine in RNA strands. So pause now and go ahead and figure that out. All right, so hopefully you came up with AUG, AAU, GUU, UGG, UAU, UAA. That is the mRNA strand. The complementary strand to that is going to be what the tRNA molecules match to the mRNA strand. So if we have AUG, the complement to that is UAC. AAU, complement is UUA. GGU, the complement is CAA. ACC, AUA, AUU, okay? Now, the next thing we have to do is figure out what amino acids that tRNA strand is going to bring to the ribosome, okay? We are going to use on our sundial chart, or on the chart that I showed you how to use in class, we're going to use the mRNA strand. So we are going to use these codons that are in green, those are the mRNA strands. We're going to use those to figure out the amino acid sequence. So I'm going to stop here for a second, pause the video, and see if you can figure it out. All right, so using that sundial chart, hopefully this is what you came up with. Yikes. Methionine, that's at the beginning of every protein. Asparagine, valine, tryptophan, tyrosine, and we have a stop codon, and that tells the ribosome that it is done. So let's look at the relationship between genes and proteins really quickly. Genes are nothing more than instructions for building proteins, okay? So what do proteins have to do with the color of a flower, or a human blood type, or dimples in a cute little baby's face? Well, everything. The traits of any organism are the result of the proteins that are being built inside of the cells. And those traits are read from the instructions, which come from, hopefully you're thinking, DNA. So, if there's a problem, on occasion, cells make mistakes in copying their DNA. An incorrect nitrogen base can be inserted, or a base could be altogether skipped. It's hard to believe the DNA polymerase, when it's making the DNA strand, it goes back and it proofreads, but it can make mistakes, especially because it reads it at such a rapid pace and it copies DNA so quickly and so many times. Mistakes can happen. Those mistakes are called mutations. And a mutation is just a change in the genetic material of a cell. Mutations can be either gene mutations or chromosomal mutations. Gene mutations produce a change within a single gene, but chromosome mutations produce changes in the whole entire chromosome. So let's look at this really fast. Why are mutations so important? Well, let me tell you. Most gene mutations, so most mutations that happen within that DNA strand, are neutral, which means they have little to no effect at all. Some mutations can cause very dramatic changes that change the normal cell function, and that is disruptive, which could then result in a genetic disorder, and that's when it becomes a problem. But some mutations could actually be beneficial, and soon we're going to start talking about evolution and natural selection and how organisms are that are better suited for their environments are going to be the ones that survive and pass their genes on. So maybe sometimes that genetic mutation could cause a trait that actually makes that organism more beneficial to its environment. Those organisms that are better suited are more likely to survive, they're more likely to reproduce, and they're more likely to pass on those favorable traits to their offspring, and we call that natural selection. Okay, so I just really quickly covered a couple of topics. I showed you how to read a amino acid chart. I talked about translation, so how a ribosome reads the mRNA strand, and that mRNA strand determines the tRNA that matches it, which determines the amino acid. And then I kind of threw in at the very end mutations and why they could be harmful or beneficial, okay? Covered a lot of material. So don't hesitate to ask me questions in class. We will review. When you are making notes, make sure that you ask questions. You write your questions down so you remember what to ask me in class. There are no silly questions, okay? Make sure you ask them in class. This is a really difficult concept, and it's something that students traditionally struggle with every year, so don't feel like you're the only one. I'll be happy to talk about anything that you want to in class. You just have to ask, okay? I hope that you got a at least small grasp on this. We will practice and practice and practice and practice. And I hope you have a great evening, and I will see you soon.